this is working. Perfect. Okay, so this is lesson 2, which is a continuation of the lecture 1 PowerPoint slides. So we left off at slide 30, and we talked about the law of segregation, which basically says that alleles get segregated into different gametes during gametogenesis or meiosis. And now we're going to talk about independent assortment. So, independent assortment is Mendel's rule that says that different alleles assort independently of one another. And so, in this case, we have to talk about at least 2 alleles. And what he used was big Y, and big R, which is yellow and round, right? So big Y is yellow and big R is round. All right, so these are seeds. And then we have, by contrast, the recessive trait, which is little y and little r. So I guess I'll go on this side. So little y is green. That's recessive for seed color. And little r is not round but wrinkled, which is the recessive trait. All right. So Mendel crossed these, right? Yellow round seeds with green wrinkle seeds, and we know that. You should know that. Like, if you're going to pass on an allele, you have to pass on at least one allele for, say, this isn't really true because there's a lot of genes involved in eye color, but let's just say there was one. You would have to pass on at least one allele for eye color and at least one allele for, say, hair color. So that's what's going on here. You have to pass on one of each. So in this case, the only option is big Y, right? For in the Y scenario, that's seed color, right? It's either yellow or green. So you have to pass on seed color and you have to pass on seed shape. And in this case, it would be big R because that's all there is. And this would be the gamete that would be formed. Right? And then you'd have this one, and all it can pass on is little y and little r. And when you do that, this is the gamete that's formed. So remember, any sperm can fertilize any egg. Oh, sorry. That's a female gamete. All right. So any sperm can fertilize any egg. And so we only have one variation of sperm and one variation of egg. And so all the offspring are going to be big Y, big R, little r. Right? And if we drew them out on chromosomes, it would look like this. We'd have one chromosome that would have color. It'd have a locus with color on it. And then we'd have another chromosome that would have a locus for shape, and that would have shape on it, right? And so if we looked at it like this, this would be big Y, this would be little y, this would be big R, this would be little r. All right. And so we're going to mate these two at sibs. So this is our P generation. This is our F1 generation. And so we cross them with their siblings, which have the same genotype because that's all we can get in the F1. And then they're going to make gametes, right? So the idea is that you can pass on any one of these. And so they could be packaged in any way. They could be packaged like this. So big Y and big R could get packaged together. So we could have a gamete like this. Or, hang on, let me just erase this. Big Y and big R could get packaged together. So let's get rid of this. Okay. We could have big Y and little r get packaged together. So the other gamete, and remember the rule is you have to pass on one. You can't pass on two Y's and no R's. Just like you can't pass on all eye color and no hair color to your offspring. All right, so let's get rid of this. Come on, there you go. And we could also have little y, little r packaged together. And the last one we could have is big R, little y. Because I already have big Y, little r. So big R, little y, big R. I'm just trying to keep it consistent. So these are the four gametes that each F1 offspring could make. Let's say that this is the male, and then the female would be able to make the same eggs. And any sperm could fertilize any egg, right? So that's what independent assortment means. And if we did the Punnett square out, you would see, and it's already done. I'm not going to redraw this whole thing, but you can see there's a quarter probability for each one of these, right, to be produced. And if you multiply 1/4 by 1/4, you get a 16th. So this constantly comes out in a ratio of 9 to 3 to 3 to 1. And Mendel showed this with lots of different traits, lots of different. This is called a dihybrid cross because there's a hybrid means it has two, right? It's a heterozygote. So we have two heterozygous for two traits, and a trihybrid would be heterozygous for three traits, and a tetrahybrid, and so on. So we're not going to get crazy right now. We're just going to stick to two because we already did one. And so this gives us a 9 to 3 to 3 to 1. If it was a monohybrid trait, then we would get that ratio of 3 to 1. These are phenotype ratios, by the way. Right, what you see. So this would be both dominant. This would be one recessive and one dominant. This would be one dominant and one recessive, and this one would be all recessive for both traits. Recessive for both traits. Whereas here, this 3 to 1 is dominant to recessive, right? So on Mendel, we did purple flowers versus white. All right, so if we look at the ratio 9 to 3 to 3 to 1, we can predict that. What's the dominant? Well, it's yellow, right here, and it's also round. So we would expect 9, and we can add these up. 9 plus 3 is 12, plus 3 is 15, plus 1 is 16. So 9/16 of these we would expect to have both dominant traits. So they would be yellow, right, and they would be round. And you could go through this and see, okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. And then we could do 3 out of 16, and that would be one dominant. So in this case, it would be yellow and wrinkled. And if we look at the yellow and wrinkled, we'll see that there's one here, one here, one here, one here, right? And then 3/16 would be one recessive and one dominant. So recessive for this would be green. And then on the other hand, the dominant would be round. So we look for how many green rounds? Well, there's 3. They're all right here. Easy. 1, 2, 3. And then the last one would be 1/16. And those would be both recessive. So they would be green and wrinkled, which is right here. This is the only one of all of these in the Punnett square. Okay, so this means traits assort independently of one another. Yellow doesn't have an influence on round, right? They don't have to go together. They can go in any order. And here's the real deal is that we have 20,000 genes. Plants probably have the same. I'm not a plant guy, but I'm going to guess. And so this would be quite different. I'm going to erase this side only. This would be quite different if they were on the same chromosome and not different chromosomes. And that's what we mean by independent assortment, because not all things assort independently. So we have 20,000 genes, and let's say the plant we're looking at has, 20 chromosomes. Well, that means every chromosome is going to have 1,000 genes. So chances are that you're going to get uh, a bunch of genes on the same chromosome. So let's look at it this way. Let's say the plant that we just crossed had them on the same chromosome. So this is the parent, right? We drew it independently over here, but let's say they're linked, right? They're on the same chromosome, so they go together. They're not independent of one another. And this would be, so this is big Y, big R, and then, right, this is just the gamete that this parent would produce, right? We're talking right here. And then this would be little y, little r, right? Which would be little y, little r. And that would be this gamete that this parent would produce, right? And so this would fertilize this. And so we have these all in one cell, right? And so this genotype of this would be big Y, little y, right? Big R here, little r here. And then we cross it with its sibling or whatever. All right, so now these are going to get packaged into gametes, but there's no way to get big Y and little r, or little y and big R, because they go together. You can't break chromosomes apart when you're undergoing meiosis. So when the gametes are being produced, these go together. So the only thing that you can make for a gamete is big Y, big R, or it could get packaged like this, where it's little y, little r. This, the ones that we have, so this one's possible. This one's possible. This one is not possible if they're linked like this, right? If they're on the same chromosome. This is also not possible, impossible if they're linked. So this is independent assortment. This is not independent assortment. And for whatever reason, somehow, we don't know, Mendel experiments always showed that they were independently assortment. So either he got really lucky because he was looking at 13 genes and they all happen to be on different chromosomes, or he cheated and threw out his data. I'm just going to put it out there, which he probably did. He probably took out stuff that he didn't fit his model. But, the guy's dead and I can't judge him. And he was dead when they found his paper, so they couldn't ask him. Anyway, it's also possible to have linked genes where you could have one chromosome is little y. I'll just erase this. We would have little y, big R, and big Y, little r. And then these gametes right here, big Y and big R, would be impossible to make because all you can make is little y, big R, or big Y, little r. You couldn't make this. All right, so this is what we call, when we talk about genes that are linked, that means they're on the same chromosome, they're linked together. Chromosomes can't explode or get chopped apart during meiosis when gametes are formed. So they go together, and they have to go together. So anyway, that's what independent assortment means. If you guys have any questions about it, let me know. But you should be able to figure this out in the problem sets. So this is what Mendel said, right? The chromosomes line up, and then they assort independently, kind of like what I showed you here. But that's not always the case. Okay, so next thing, what happened to draw? What is you? What, All right, so probability statistics, and there's some of this on your problem sets as well. So I just want to remind you, right, when we talked about gametes, right, like if it's big P, little p, right, there's 50% chance of each of these being made, right? It's the same thing as tossing a coin and having it come up heads or tails. This is supposed to be a big P, and this is supposed to be a little p, just so you know. But they look similar. But that's what it's trying to say. So every one of these has a 1/2 chance, and every one of these has a 1/2 chance. So if you're flipping two coins simultaneously, the odds of getting two heads is 1/2 times 1/2, which is 1/4, right? And that's the rule. That's the rule of multiplication. Okay, and uh, this one would also be 1/4, and the same thing here, and the same thing here, right? So they're all 1/4. However, these are the same, right? It doesn't matter if it's heads or tails or heads. You flip it simultaneously, these are the same outcomes. And because they're the same, you can add them together. So, for example, big P, or little p, big P, they're the same uh, outcome. They're both heterozygous. So, if they're each have half or a quarter probability, this is just for gametes, right? Not for the offspring. So if we did the Punnett square like this, and we had big P, little p, and big P, little p, we're doing it out. Each of these would have a 1/4 chance of occurring. And so this probability would be 1/4, and so would this one. But we can add those two together, and I already kind of went over this, which is 1/2, right? And so in this case, we have a 1 to 2 to 1 ratio. 1 is big P, 2 is big P, and 1 is little p, little p. Right? And this we came up from the rule of addition. And all these probabilities came up with the rule of multiplication. So hopefully you guys get the difference between the rule of addition and multiplication. It's the same thing with dice. So let's say that we were going to do dice. Okay, this is good. This is a good one. Here we go. This is great. Let's do this. Mm-hmm. Okay, here's our die chart. I know it's kind of small, but hopefully you can see it. So, what's the probability of rolling a 1 on a 6-sided dice? Well, it's 1 out of 6. Just like what's the probability of flipping a head or a tail? It's 1/2 because there's only two outcomes. When you roll a 6-sided dice, there's 6 outcomes. If you rolled a 20-sided dice, there's 20 outcomes. if you're talking about uh, purple versus white, there's only two outcomes, right? So it's each of those is a half. If you're talking about like throw another trait in there, like purple flowers versus white or white flowers and tall versus short plants, then the probability changes. Now you have four possible outcomes instead of two, and so the probability of having any one of those one outcomes is 1/4. Just like rolling a six-sided dice is 1/6, or tossing a two-sided coin is 1/2. So that's the rule of multiplication, right? If we throw another dice in there, then it's 1/6 times 1/6 or 1/36th, right? Um, so, I mean, I don't know if you play craps or not, but this is actually exactly how they calculate craps odds. And you could be a better craps player if you realize this. Okay, there's only one way to get a two, right? The only way to roll two dice and get a two is snake eyes, two ones. Can't get it any other way. So the probability of rolling that is super low. It's 1 out of 36. But there's different ways to get a three. You could get a one and a two, or a two and a one, right? So each of these probabilities is 1/6 times 1/6, 1/36. I wonder if it'll let me write on here. I'm going to try. No, I don't want to highlight. I want to draw. Okay. So this one here, right? Let me draw. Come on. This one here. Yes. Right there. Right. The odds of rolling a 1 is 1 out of 6. The odds of rolling a 2 is also 1 out of 6. Any number on there is 1 out of 6. So 1 out of 6 times, my computer is lagging here. 1/6 is 1/36. Oh, what just happened? That's okay. Equals, no, what are you doing? It's 1/36. It's really big, but that's okay, right? And then this one is also 1/36. And since it's also a 3, we can add those together. I'm going to exit this. We can add those together and it becomes 2/36, right? Let me try to make this bigger. So this is 1/36, right? The rule of addition says we can add threes together because they're the same outcome. So we have 2/36. The odds of rolling a 4, well, there's three ways to roll a 4. A 1 and a 3, a 3 and a 1, or two twos. So we add 1/36, 1/36, 1/36. We get that from the rule of multiplication. Rule of addition says we add them all together. So it's 3/36 probability of rolling a 4 with two dice. To roll a 5, there's 4 possible outcomes. To roll a 6, there's 5. To roll a 7, there's 6. And then we go down from there. So 8, there's 5. 9, there's 4, 3, 2, and 1. So that is a great example of the rule of addition and the rule of multiplication. Um, and it comes in super handy if you want to bet odds on craps. What do you think the most what's the highest probability that you're going to roll? 7. What's the worst bet? 2 or 12. And 11's not that great either, just so you know. All right. So we're going to do a lot of statistics in this class, and I just want to make sure you guys understand the rule of addition and the rule of multiplication, because we're going to use that to predict the outcomes of offspring. Okay, so I want you guys to work. I usually do this in the class, but since this isn't online, we're just going to do it. So I can do this relatively quickly, and I'll tell you why. I know the probabilities. I have them memorized. If you wanted to, you could draw Punnett square, and it would take you forever on the test. But I know that this is a monohybrid cross, right? And this might be better. No, it's not. So that's a monohybrid cross, right? In a monohybrid cross, I instantly know the ratio is 3 to 1 for phenotype and 1, don't like that color, 1 to 2 to 1 for the genotype. So phenotype is 3 to 1. In Mendel's world, purple flowers, purple or white. So 3 out of 4 are going to be purple, and 1 out of 4 are going to be white. And this is the genotype, right? So I know 1 out of 4, right? 1 plus 2 plus 1 is 4. So 1/4 are going to be both dominant. So big P, big P. 2 out of 4 are going to be 1 dominant, 1 recessive. And then 1 out of 4 are going to be both recessive, homozygous, recessive, homozygous, dominant, heterozygous. So those are the frequencies. So now, knowing these frequencies, I can super easily answer this. What are the odds of getting a big P, little p genotype? Well, 1/2, right? Actually, it's 2/4. I just reduced it. 2, remember this is out of 4. 1 out of 4, 1 out of 4. All right. What are the odds of getting the little p genotype? Oh, well, again, super easy. It's 1/4, right? I know that because I know the ratio. What are the odds of getting a purple flower? Purple, right? It's super easy. It's 3/4, right? So the answer to this, purple is 3/4. What are the odds of getting a big P, little p? It's 1/2. What's the odds of getting a little p is 1/4, right? Just because I already know the ratios. I've memorized them. And I strongly suggest you do too, unless you love, love, love drawing Punnett squares. And then that's on you, which is really odd. Okay, what type of cross is this called? It's a monohybrid cross. What are the odds of getting a wrinkled green seed from this cross? Okay, well, I already know the ratios. It's 9 to 3 to 3 to 1, right? Because it's a dihybrid cross. This is a hybrid. This is a hybrid. Hybrid is the same as heterozygote. So di, 1, 2 positions. Dihybrid cross. Always get 9 to 3 to 3 to 1, right? If it's independently assorting, and that we assume that it is. Okay, what's the odds of getting a wrinkled green seed? Okay, wrinkled I know is recessive. Green I also know is recessive. So that is this category, which is, remember we add them all together, that's 16. So it's 1/16. What are the odds of getting a round green seed? Okay, round is dominant and green is recessive. So it's got to be one of these, which are the same. So it's 3/16. What are the odds of getting the big R, big R, big Y genotype? Okay, so here we're not talking about phenotype anymore. We're talking about genotype. And I'm going to show you a little trick to figure this out relatively quickly. Remember, because these are independent of one another, we can calculate them independently of one another. That means I know the genotypic ratio of a monohybrid cross is 1 to 2 to 1, right? We said that right here. So this cross is going to be the same. It's a monohybrid cross. So the outcome of getting a big R right here is 1/4. So the outcome of getting a big R from this cross is 1/4. Now the outcome of getting a big Y, remember I can make them independent of each other. I don't care about R anymore. All I care about is Y. I'm trying to fix this. Okay, so I know the outcome of this to get a big Y is also 1/4, right? Both dominant right here is 1 to 2 to 1. So now I can use the rule of multiplication, and that tells me that the odds of getting big R, big Y is 1/16. And if you don't believe it, we can go back and look at the actual cross, which I believe is here, here, here. Yes. So big R, big Y is 1 out of 16. There's no other outcome for this at all. All right. So on a test, like I could ask you anything. I could ask you, remember you got to know the ratios to answer this. But I could ask you, what's the probability of this cross? Let me use something else. Let's use X, Y, and Z. X, X, Y, Y, and Z. Z cross to X, X. These are little X's. Um, Big Y, little Y, and then we'll make these both big Z's. All right. So I could make a Punnett square of this, and it would be a pretty big Punnett square. But I know that the cross of this times this is going to be I forgot to tell you what the offspring is. So the offspring, I want to know what the probability is that they're going to have this. This offspring. Right? So, okay, so let's go. So we have little x, big X, little x, little x, little x. I want to know little x. So I know that that's 1/2, right? 1/4 plus 1/4 is a half. Now I cross these, right? So let's get rid of this. And we have big Y, Punnett square, and then we have big Y, big Y, little y. So we have big Y, big Y, big Y, big Y, big Y, little y. So again, this is 1/2. And then we can do this same thing, and I can tell you once again, it's 1/2. So the odds of having this genotype is 1/2 times 1/2, which is 1/4, times 1/2, which is 1/8. All right, and you could do some other practice problems with this too. I don't want to spend a lot of time on doing practice problems for this, but let's just say I asked you on the test. We'll do X, Y, and Z, X, X, Y, Y, Z, Z, W, W, cross to X, X, Y, Y, Z, Z, W, W. And then what's the probability that they would have this offspring? So all these are dihybrid crosses. I already know the ratio. The probability of getting this is 1/4, 1/4, 1/4, 1/4. So 1/4 times 1/4 is 16, 64, 2, 1 out of 256 that they would have that. Or you could make a Punnett square that's 250 squares big. Which one would you rather do? I was just saying. Okay, so that's really all you need to know for Mendelian genetics, right? We're going to get deeper into this. We're going to talk about linkages and how we can use those to figure out map distances and how close genes are and where their locus is and all that other stuff. And then the probabilities of having certain outcomes of offspring, because that's really important too, right? I mean, like if you looked at, I think there's a Lifetime movie called My Daughter's Keeper or something, but there is a incident where uh, a child had leukemia and needed a bone marrow transplant, and they could not find a match to that kid. And so the parents went to a genetic counselor and said, what's the probability if we have another kid that it would have a matching uh, profile to the our daughter's immune system, right? And it turns out it's a simple dominance thing. So the answer was, and she was homozygous recessive for that. So the answer would be 1/4, right? You know that from the 1 to 2 to 1 ratio. So they decided to have the kid, and they got lucky, and the other kids they used the other kid's bone marrow to save the life of their first child. Um, Now, if they went to the genetic counselor and the genetic counselor said there's 1 in 62 million, they might not have really had another kid to try to save her because the chances are really super slim. Probably better to buy a lottery ticket. So, genetic counselors do a lot of stuff like that. It's a really important job. And you get paid pretty well, right? Like over 6 figures. So, understanding the outcomes and the probabilities are critical. I mean, some people spend millions of dollars on horse genetics, right? You want a horse that has good genetics, that's fast, you know, hundreds of millions of dollars. So this dogs are outrageously expensive these days. So there's a lot of like just, breeders, horticulturists, you know, animal breeders, farmers, what have you, that need geneticists to tell them what works, what doesn't, what's the probable outcomes. All right, so these are exceptions to Mendel's rules. Mendel only wrote about complete dominance, right? And that means that one thing is completely dominant over the other. So in our case, it's purple or white, right? Um, but that's not always the case. Remember, I said that this is the default, so it just defaults to white. If there's no purple pigment made, there's no pigment made at all. The default is white, just like your wall. Well, now imagine, okay, you have red, right? In this case, this is the snapdragons. You have red, and then you have white. Well, they're not, one's not dominant over it, so we don't use this nomenclature anymore. We just say the color is red, right? And you could have two, remember, these are just representative of chromosomes inside the parent. So they would have two red copies that would make a protein that's folds and reflects light in the red wavelength so that you see it, it's red. This would be the one parent, and then the other parent would be white. So we don't use like little r or whatever. We use CW since they're not dominant. They're incompletely dominant. And so again, this is representing just chromosomes that are white and white. Right? And they're going to make a product that actually it's a real product that is really white, right? So it's going to make a protein that reflects all of the colors of the rainbow, which is what white is, right? Roy G. Biv. And so that flower appears white. It's not a default pathway, it's actually a pigment. So what do you think happens when you mix red? I'm mixing these up. What happens if you mix white with red? Well, you can go home and do it. You put it together and you'll see that you get pink, right? Now, if white was a default color and red wasn't present, then, or if only one red was present, it would be red. So that's the difference between complete dominance and incomplete dominance. Is that in complete dominance, one is completely dominant over the other, and incomplete dominance, they share dominance, right? They are a blend of the two traits combined. This is a little tricky for genetic students because there's another thing that we talk about. Oh, and by the way, this ratio is the same as the genotypic ratio. So 1 to 2 to 1 ratio. So if you see that, a phenotypic ratio of 1 to 2 to 1, you can think incomplete dominance. If it were complete dominance, what would be the ratio you would expect? Correct, 3 to 1. All right. Yeah. So there's another thing called codominance. A lot of students are like, okay, well, what's the difference between incomplete dominance and codominance? On a molecular level, there isn't any. But on a phenotypic level, there is. And so the terms are different because one is blending of the two together. Like on a molecular level, you could probably get down to the point where red and white molecules are present, and so they're just causing it to be pink. But think of it as just kind of coming together and making one. And that's what incomplete dominance is. Codominance is that both are expressed simultaneously. And so one example that they use for this is blood groups. So in a cell, right? And everybody has a blood type. In your cell, you have receptors. Hopefully you learned this in intro biology for majors, 181. But you have a cell, and in that cell, you're going to have receptors. So there's going to be alleles, right? And those alleles are going to be for blood groups, right? So you could have an A allele, and that makes an A receptor. I shouldn't say it's an A receptor, not a receptor, but a receptor. So let's say A receptor has a shape of a Y, and that means molecules that are round are going to fit in and bind to that. And then you have a B allele. So remember, these are multiple alleles. So we're just talking about one chromosome, multiple alleles. B, it makes the B receptor. And let's say that B receptor is shaped like a goalpost, right? So in this case, a square object would fit in that better than a round object. Or you could have an O, which means you make no receptor. And I don't have to draw that because there isn't any receptor. All right, so this is the possibility. Now, you have two alleles for every trait. This is your ABO blood group trait. So you could be AA or AB or AO or BO or OO. I miss anything? I mean, you could be BA, but that's the same. Oh, BB. So those are the possible outcomes, right? So if you look at a BB cell, it just has twice the number of goalpost receptors on it so let's say it has 6 as opposed to a BO, which would only have 3. But the receptors are the same and they're the same shape. So if you're AA, your blood type is going to be A because you have two A's. If you're AB, your blood type is going to be AB. If you're AO, well, you have one. That's enough to make you A because that's just telling you what your antibodies are. BO is going to be B. OO is going to be O. And BB is going to be B. All right. So this is what it's showing you here. AA is A. AO. So it's showing you O right here. AO is also A. And then you're going to make antibodies, right? So you want to make antibodies to stuff that's not you, right? Because if you made antibodies to you, they would attack you, and that would be bad. So you're going to make antibodies to what you're not. So in this case, the A is this shape. You don't want to make circular antibodies because it's going to bind to A and kill you. So you want to make antibodies that are square shaped, right? They're B antibodies because you don't have B in you, so you're not worried about it. That's not you, right? And so if you had put B blood in your body, your B antibodies, your square antibodies would attack it and cause your blood to coagulate. In fact, any B would cause that. O has no receptors at all. So it doesn't matter what's out here, it's never going to bind to it. So you're safe, right? And then A, obviously, you're going to have A receptors, so the squares aren't going to fit into the circle receptors. So anyway, B is the same way. So BBBO, you're going to make antibodies to A because you don't have A, and then anything with A in it, you're going to get your blood coagulated, and it will kill you. So either B or O, you're safe, right? Same reasoning. AB, you make both A and B receptors. So you don't want to make any antibodies because any antibodies would either A or B would kill you. So you have no antibodies. And you're safe taking anyone's blood, right? Because um, you are not going to make any antibodies. So it wouldn't matter whose blood you have. If you have this shape receptor or this shape receptor or a combination of the receptors or no receptors. So in that, because of that, AB is considered a universal acceptor. If you have AB, you can take anybody's blood. O, you're going to be OO. You're going to make no receptors, but you're going to make both antibodies, right? And so you can't take anyone's blood that would coagulate and kill you except O, right? Only O. But because you have no receptors, you could give your blood to anyone because no matter what antibodies they have, it's never going to bind because there's no receptors. So O is the universal donor. And if you are O blood and you've given blood before, you know they're like calling you up every second. Uh, can you get blood? I just gave it like 2 days ago. Well, that's okay. Well, you'll be fine, right? And so because they can give that your blood to anyone. So that another term that we talk about is multiple alleles, right? That just simply means that you have many alleles for a trait. This is you. I mean, this isn't rocket science here. Like if you look at eye color, right? It could be brown or blue or hazel or green or whatever. So that's multiple alleles for one trait. Same thing here. You could be A, B, or O allele. So that's more than two. Um, Same thing with hair color or whatever. So a lot of traits are like that. So blood groups are an example of codominance, right? Which means that they're both expressed if they're present. A and B are found on the same cell. A and B expressed simultaneously in the same cell. They're not mixed together. It's not something like this receptor, right? So because that would be what they would consider um, incomplete dominance, not codominance. I know it's hard to think about, but that's kind of what geneticists mean. I don't think they really meant on a like a super small molecular level when they're naming this stuff, even though this is a pretty small molecular level. All right. So any questions about that? Hopefully no. And then let's see. We're going to go to another genetic term, pleiotropy. So pleiotropy is a term, a genetic term you're going to need to know. So just get a note card out or however you need to memorize this. And that means that a single gene, a single mutation that causes a phenotypic trait, like a disease trait, uh, has multiple phenotypic effects, right? So let's say that in this case the gene for this is a mutation in the hemoglobin gene. So a mutation in the hemoglobin gene causes you to not be able to carry oxygen properly. Your cells collapse and they make a sickle shape, right? So you can't carry as much oxygen in this form, right? And so you're anemic. And that's why it's called sickle cell anemia. But there's also other consequences, right? Your blood vessels are like this. And so having nice round blood vessels or round blood cells they're just gonna roll their way through your blood vessels no problem, right? But if you have sickle shaped blood vessels, they're gonna like fly around here and like catch their sickle into your cell wall. And then that's gonna clog up your blood vessels. So you get blood vessels clogging your arteries. You could get it could cause pain. You could get fever. It could cause brain damage because you're not getting enough oxygen. You could get heart failure, right? And then the red blood cells not carrying oxygen or anemic that make you tired. Uh, and then this can also you can get clumping in your spleen that can lead to damage of your spleen, damage to other organisms, and then so you get impaired mental function, paralysis, pneumonia, rheumatoid arthritis or rheumatism, and then kidney failure. So because there's multiple phenotypic effects here and here from one single gene or mutation in a gene, uh, that term is pleiotropy, right? And that happens a lot in diseases as well. All right, can I move this? Okay. So, more genetic concepts. So, epistasis means that two or more genes are interacting in a pathway. So, this is pretty common. If we look at something like Roundup, which you may have bought at the store, and you spray it on your weeds and they die, right? And you don't really care how it works, but I do, because it's a multi-billion-dollar biotech business by Monsanto, and so I'm always interested when it has to do with genetics, molecular biology, or money. So we have A, right? It's like let's say this A product, and the way that this works is that plants. I don't know if you should know this. So plants do photosynthesis, right? This only works on plants. When plants are doing photosynthesis, the only thing that they can make are carbs, right? They can't make proteins. If you remember photosynthesis, the output is glyceraldehyde 3 phosphate, and that is one half of a glucose, which is a carb, right? And plants link those together and they make glucose. Long chains of glucose are called carbs, then your nutritional label. And so that's all plants make. So they have carbs, lots of carbs, but they also need to make proteins, E, and to do that they need amino acids. Right? So this is actually an amino acid pathway. So you start with carbs and you end up with amino and carboxylic acid. I forgot a hydrogen in there. Yeah. And OH, and then let's just make it simple. H, H. Oh, actually, this is a carbon, not hydrogen. Carbon. There you go. Okay. So this is the amino acid. And you just do this by, you're simply replacing uh, carbons, adding nitrogens, and rearranging this molecule. And to do that, you need enzymes. So there's actually four enzymes in this pathway. One enzyme, we'll call it small a, that produces B, right, product. And then enzyme, we'll call it B, produces C product. And enzyme C produces D product. I'm running out of room. And then enzyme D produces E product. So we have 5 products, 4 genes to convert carbs into proteins. And when you spray your Roundup in there, it inhibits, this is called glyphosate. Not that you care, but anyway. And that inhibits a gene in this pathway that blocks it. And so it would be like if you had a iPhone factory and the people that put the chips in didn't show up that day. What would happen to your plant? It would shut down. So if it shuts down, plants can't make proteins. What happens to them? They die, right? So that's an example of epistasis, right? It's multiple genes working in a pathway. And a common way to learn this is looking at like either dogs, like labs, or mice. So we have 2 genes here. One, oh, let's start over. Okay, we have uh, B gene, right? And that in the dominant form, that allele produces black. And in the recessive form, it produces brown. Right? And then we have a C gene, and C gene means you have to have C to put the pigment, put this pigment, whatever that is, put pigment into your skin, into your hair, and hair. And little c is broken, so it doesn't put pigment into the hair. Okay, so this is a standard cross. This is a dihybrid cross, right? So we would expect a 9 to 3 to 3 to 1 ratio. But as you can see, we don't get that. We get a 9 to 3 to 4 ratio. And that's because of these 2 genes working together. So in epistasis, usually you get these collapse together, so you get a 9 to 7 ratio. What did I just do? 9 to 7 ratio. Or maybe these 2 collapse together and you get a 9 to 3 to 4 ratio. But if you see these ratios in offspring, you 100% could say, oh, those are genes working together in a pathway. And so here's how it works, right? So this mouse can make big B, big C, little big C, big B, and so on, just whatever. This is the egg, so this must be the female. And this is the male, so it's making sperm. Any sperm can fertilize any egg because these are independent of one another. So they're independently assorting. And this is our outcome. So big B, that's black. 2 painters, right? 2 genes to put this pigment into the skin. What color is the mouse? Black. Easy. 1 black and 1 brown. Well, black's dominant to brown, so it doesn't matter. It's still going to be black. 2 painters, yeah, that's going to put black in the hair. That mouse is black. This is going to be black, right? Black is dominant to brown. 2 painters, black. Black, 1 painter, that's okay. You don't need 2 painters. It just takes longer. You still get the same color. This one is black, 1 painter. Okay, still going to get the same outcome. Black, heterozygous, still black. Heterozygous for painter. You only need 1 copy of black and 1 painter, and you're black. Same here and here. This one, 2 little b's means you're brown, right? This is dominant to this. So, and then you have 2 painters, so for sure the brown's going to get put in your skin. This one, brown, 1 painter, still. This one is brown, 1 painter, still works. Okay, here. In these white mice, we have black. Black is made, but nobody's there to paint it. It would be like if you bought paint and Never painted it on your wall. What color is your wall going to be? Still going to be white. So this mouse is white. This one would be black pigment. No painters. Mouse is white. This is brown. Still no painters. So this is white, and this is black. No painters. It is white. So we get a 9, right? Black, 3 brown, 1 or 4 white ratio. So 9 to 3 to 4. So I see that. I know that's epistasis. I might have to work out the genetics to figure out which of these is the pathway, what's going on here. But at least I would know that there's genes working together, right? Because of the ratio outcome. And that's a good starting place for geneticists to then decipher what are these genes in this pathway. We're going to talk more about epistasis in detail in future lectures, but for now, that's really all you need to know. And by the way, this is the same thing that happens with labs, right? That's why you have the 3 flavors of Labrador Retrievers. You have black, brown, and yellow. And it's because of this exact pathway. All right, last slide. Polygenic. So poly means many, genic is genes. So polygenic inheritance means several genes determining a single phenotypic trait. Eye color is an example. If you look at your eyes, they're not just one color. If you look closely at your eyes, there's lots of colors in your eyes. So there's lots of genes that make up eye color. It's a myth that eye color is a single gene, right? I'm just telling you as a geneticist, I know that for a fact. But here's an easier thing to recognize and remember is that skin pigment. So in humans, we know that there are 3 genes, at least 3, there may be more, but at least 3 genes. And that means so that's 6 alleles, right? Because every gene has to have 2 alleles on the chromosome. And we're going to call it A, could be AA, and then we'll call this B, could be BB, right? This could be a parent, and then C. Actually, let's do this. We'll do the heterozygote. So AA, B, little b. Well, you can't really tell much about the difference between this, but I'll make that C, big C. All right, so this is what one of the parents would look like, right? And each of these alleles produces a specific amount of melanin, right? That makes your skin dark. Not melatonin, that makes you sleepy. Melanin, so this has 1, 2, 3 units, right, out of 6 possible units. So you would think that this person would be roughly in the middle, and you would be right as far as skin color, right? 3 units is right in the middle. And again, this is the same as the dice throwing, except here we're doing 3 dice, right? So it's 1/2. The possible number of gametes for this is, all right. I should go back and do this. I think I did this, but maybe I didn't. So the number of gametes is determined by the number of traits, right? And this is for heterozygotes. So if you have 1 trait, like for the P's, like big P, little p, then the number of gametes that you can produce from this is 2. 2 to the first equals 2. If you have this, right, like yellow and wrinkled around, right, then that is 2 to the second, which is 4. 2 times 2 is 4. Gametes that you make, right? And we could go back and look at this. Too far here. So gametes 1, 2, 3, 4 gametes, right? Because it's 2 traits. So this would be 2 to the second number of gametes. All right. So let's say if it's 3 traits, right? So 3 traits, like in this case, AA, BB, CC, that would be 2 to the third, which is 2 times 2, which is 4 times 2, which is 8, right? And if you did a Punnett square, because there's 2 parents, it's 8. So every one of these is 1/8, 1/8, right? And then the rule of multiplication says we multiply those together. So 1/8 times 1/8 equals 1/64. So you could get every one of these Punnett square things. I'm not going to draw an 8 by 8 Punnett square, but you know that it has 64 squares. So every genotype outcome is 64. And so this is saying if we added all of them together, 20 out of 64 would have 3 copies in any order, right? 3 pigment copies. It could be Big B over here or whatever. If we did this, then you would get 0, right? And if we did this, you could get all 6. So two heterozygous parents for each one of these alleles could have this range. And this range we call quantitative genetics. And there's a whole chapter on this. Quantitative, because we're going to quantify what the probability is for each one of these in this range. And then what's going to happen to the next F2 generation and so F3 generation and so on. Okay, so polygenic means multiple genes are influencing that inheritance. And then I'm going to pause this for a second. Okay, I didn't want to waste time looking this up, but I found it. And so here's a perfect example of this. So both of these kids' parents are heterozygous for those 3 alleles. And so these are twins, right? They're not identical twins. Their mother released 2 eggs and were fertilized by 2 separate sperms at the same time. They were born at the same time. And this is what they look like, right? So if you said that they were sisters, people would probably say that's BS. But on a genetic aspect of this is completely possible. And honestly, she could have a lot darker skin. She could have a little lighter skin. So theoretically, this is possible. And so, it makes me laugh whenever I see, Maury or Jerry Springer or any of these like things that test, are you the baby's daddy? Doing that, and the first answer the dad always has is, that's not my baby. And the reason is it looks nothing like me. But that doesn't matter, right? Because genetics is complicated and you can get anything except maybe a dog. If it was a dog, then you'd be like, yeah, I'm not the baby daddy. All right. So anyway, I'm going to stop this lecture now. That finishes the first, so I'm going to call it a lesson. So this would be the second lesson for the first lecture series, which is on Mendelian genetics and variation of Mendelian genetics. You guys should, once you finish this, you can answer some of the questions in the study guide, obviously, for exam 1. You also should be able to do the quiz for the lecture 1, which is in there, and also you need to do the syllabus quiz. For whatever reason, there's a bunch of drama about people signing up for classes and never showing up and taking the student loan money and scamming the government. So I have to make sure that you guys are showing up to class and actually participating the first week. So if you haven't done that, you need to do that. I'll send out an announcement in an email as well. I also wanted to let you know that I updated the syllabus. So I included quizzes in there to be 5% of your grade, which I originally didn't do. But this is kind of a gift, and you guys should thank me because I'm going to let you take the quizzes 3 times and take your highest score. So no way should you, at this level, not be able to ace this quiz. So there's going to be quizzes for most of the lectures series. I've already got the first three up. You can take them whenever you want. And I will start recording the next lecture series either this weekend or next week. So, anyway, if you have any questions, make sure you email me. Have a good one.